Hello, everyone, and welcome to the Biosynapse podcast. Today, we're diving into a really fascinating new research article. It's titled Multimodal Deep Learning for Allergenic Proteins Prediction, and it was published in BMC Biology by Lejeune Yu, Yuxin Luo, Shiki Wu, Sichen Luxie Runyu Jing, and Jiezi Luo, along with their colleagues. So, let's set the scene a bit. Allergies, right? They're a growing global health challenge, impacting millions and millions worldwide. But here's the thing, maybe the real kicker. It's apparently practically impossible and just incredibly expensive to experimentally test every single potential allergen out there. Think about that, just the sheer scale. So what if we could use cutting-edge AI to maybe precisely identify these hidden triggers, cut through all that noise and expense?
That is exactly the challenge this research tackles, and that's our mission today, really. To unpack how this new approach, they call it Multimodal-AlgPro, acts like a super smart detective. It uses advanced deep learning to predict these allergenic proteins with pretty unprecedented accuracy. It pulls insights from places we just couldn't really access before, offering a real shortcut maybe to understanding this complex field and its critical implications.
Okay, Multimodal-AlgPro. It sounds like it's definitely aiming high, trying to solve a big problem.
Yeah.
But maybe first, for those of us who might need a quick refresher, what exactly is an allergy? And why is identifying these specific triggers just so vital?
Right. Simply put, an allergy happens when your immune system kind of overreacts to something that's usually perfectly harmless. Things like pollen, maybe a specific food, even certain medications. And this overreaction, it leads to chronic inflammation. And like you said, it's a really significant and, frankly, ever-growing global health burden. So pinpointing the exact causative allergens is absolutely critical. It lets us prevent re-exposure and intervene effectively in allergic reactions, which is especially challenging when the allergens are rare or unknown or maybe hidden in these increasingly complex products we see now, modified foods, pharmaceuticals, cosmetics.
It sounds like a constant game of catch-up.
Yeah.
So given how crucial this is, what were the big hurdles, like the conceptual hurdles maybe in earlier attempts to predict these allergens? What did we try and why did it fall short?
Well, the initial approaches were, I suppose, quite straightforward. You had direct experimental testing, of course, but the sheer scale, the number of potential allergens just makes that impractical and incredibly costly. You simply cannot test everything. So researchers, naturally, turned to computational methods. Early on, we had something called sequence alignment.
Okay.
This was basically like comparing a new protein's recipe, its genetic code, to known allergens, looking for similar patterns. It was definitely a step forward, but it had a major issue, a really high rate of false alarms or false positives. And it just wasn't efficient enough for large-scale predictions. It wasn't scalable.
Right. Okay. False alarms, not scalable. And then after that, traditional machine learning came onto the scene, which we hear so much about, pattern recognition. How did that change things? And what were its limitations?
Traditional machine learning certainly improved things, yeah. These systems were designed to find connections, patterns between specific features of allergens and their properties. But here's the catch. They relied heavily on what we call expert-derived features.
Okay, what does that mean exactly?
Imagine like a tailor making bespoke suits, but for each piece of data, meticulously handcrafting every single detail based on what experts thought was important. This manual process, it meant they weren't really scalable either, and they often missed crucial sort of hidden interactions within the data itself.
I see.
Which led to problems with accuracy and generalizability. They struggled to predict new unseen allergens reliably.
So good at what they were specifically designed for, maybe, but not adaptable enough to really grasp the underlying complexity.
Yeah.
Okay, that brings us to deep learning then, which, as we know, has revolutionized so many fields. What did it bring to the table for allergy prediction?
Deep learning brought a huge advantage. It could automate that feature extraction.
Okay.
It's like our AI tailor suddenly learning to design its own patterns right from scratch, directly from the raw data. This removed the bottleneck of manual engineering, and it definitely boosted predictive accuracy. However, they had their own Achilles heel, you could say. They often need massive amounts of data to train effectively.
Right. And allergy data might not always be massive.
Exactly. In fields like allergen prediction, data sets can be smaller, relatively speaking. And with smaller data sets, these models can overfit.
Overfit. Like memorize the test.
Precisely. It's like a student memorizing answers instead of truly understanding the subject. This makes them less reliable in real-world scenarios. And crucially, many early deep learning approaches also neglected something called evolutionary conservation.
Evolutionary conservation. how proteins change over time.
Or don't change, exactly, how they're conserved across vast stretches of deep time. And that information is actually a vital clue to their fundamental properties, including their allergenic potential.
Ah, okay. So that's the gap. That's the gap Multimodal-AlgPro is trying to bridge. It sounds like it's trying to learn from all these past attempts and like pull every relevant piece of information together, a real deep dive into the data itself.
Exactly right. The urgent need for better, more reliable allergen prediction, combined with the shortcomings of the existing methods, highlighted this significant research challenge. So Multimodal-AlgPro seeks to bridge this gap by combining all these different types of data sequence, composition, those physicochemical properties, and especially that evolutionary information in a way that's robust and hopefully more accurate.
Okay, so let's get into it. How did our super smart detective Multimodal-AlgPro actually build its case? You said it's not just one clue, but a whole toolkit working together. Let's peek inside its mind, so to speak. How does it process all this info?
Absolutely. So Multimodal-AlgPro is built on what they describe as a deep learning model based on multimodal data fusion and ensemble model strategies. Bit of a mouthful, I know.
Huh.
But basically it has two main analytical brains or components working together. One is a CNN, a convolutional neural network. These are excellent at analyzing sequence data. Think of it like it's scrutinizing a protein's genetic code, word by word, right?
Wow.
Identifying specific patterns or motifs.
Okay, the sequence part. And the other.
The other is an MLP, a multi-layer perceptron. This one handles what we call molecular features, things like the protein's physical and chemical properties, size, charge, hydrophobicity, that sort of thing.
Got it. CNN for sequence, MLP for properties. And how do these different analytical parts actually work together? Is it sequential or
Well, the crucial part is what they call early fusion.
early fusion?
Yeah, the insights from both the CNN analyzing the sequence and the MLP looking at features, they're merged very early on in the process. This creates a kind of unified protein representation, almost like a comprehensive profile of the protein that incorporates all these different aspects right from the start. And this early combination, they found, significantly enhances both the accuracy and, importantly, the ability to interpret why the model makes a certain prediction. It's not just a black box.
That's interesting. So it helps understand the why, too. Okay, but before combining them, did they test the individual parts? Like auditioning the players before putting the team together?
Precisely. That's a good analogy. They rigorously evaluated 14 individual or unimodal deep learning models first. And what they found was quite interesting, actually. The CNN model, the sequence one, was surprisingly effective on its own.
Oh.
Yeah, which suggests that sometimes, simpler architectures can be highly effective, maybe, especially when you have limited data. And for the molecular features, the models that used PSSM-based features.
PSSM, that's the evolutionary information again?
Exactly. Those models, the ones capturing that crucial evolutionary information, were the top performers for those feature sets. It really underscores the importance of that evolutionary context in understanding protein function and allergenicity.
Okay, so they knew the strengths of the individual players. Then came the big challenge.
Yeah.
Combining them optimally. I think you mentioned there were, over 2,000 possibilities? How on earth did they figure out the absolute best combination? That sounds huge.
It was a monumental task, absolutely. With 14 unimodal models, figuring out the best combination from 2047 possibilities wasn't just about, throwing everything together and hoping for the best. They used what's called an exhaustive search strategy.
Meaning they tried everything.
Pretty much.
Okay.
Systematically trying out every unique combination to find the winning team. It's like meticulously trying every single key in a giant key ring to find the one that unlocks the highest accuracy.
Wow. Okay, dedication. And what was that winning combination? What did the dream team look like?
Well, the best performing combination turned out to be a powerful mix. It involved the sequence analysis using the CNN combined with multiple quite sophisticated ways of encoding that evolutionary information from the PSSM.
Multiple ways.
Yeah, think of these as different angles or lenses through which the AI examines how proteins have changed or stayed the same across deep evolutionary time, each one providing a unique piece of the puzzle to our detective.
Okay, a multi-pronged attack on the evolutionary data plus the sequence. And did this complex combo pay off? Was it worth that massive, exhaustive search?
Oh, absolutely. And this is where it gets really interesting, I think. This best combination achieved an impressive 93.1% accuracy.
93.1, that's high.
It is. But here's the real aha moment, maybe. An astonishing 1937 out of the 2047 multimodal combinations they tested outperformed even the very best single data source model.
Whoa, wait. Almost all of them. Like over 94% of the combinations beat the best single approach.
Exactly. It's not just a win for allergy prediction. It's a powerful demonstration that in complex biological systems, integrating diverse data streams isn't just, a small incremental improvement. It often leads to these kinds of synergistic gains that single source models just can't achieve. It kind of shifts our thinking from find the best individual feature to how do we best combine all the relevant features.
That's a remarkable finding. Truly remarkable. Okay, so it predicts well, but you also mentioned interpretability, understanding the why. Can the model actually help us see why it thinks something is an allergen?
Yes, and that's where multimodal interpretability comes in, as they call it. The model doesn't just give you a yes-no answer, it helps you understand the patterns it's seeing. They use a technique called UMAP, which creates these clear visual clusters or neighborhoods for allergens and non-allergens.
Like a map.
Sort of, yeah. A map showing how the AI groups these proteins. This visually confirms that the model is genuinely learning to differentiate these protein classes based on distinct features it's picking up. It's not random.
Okay, so UMAP gives the big picture, showing it can separate them. What about the specifics? Can it tell us which features are most important?
It can. Using another technique called SHA, they could analyze the contributions of different features. On a global level, sort of looking overall, they saw that the sequence data processed by the CNN had the biggest influence. That makes sense. But the evolutionary information, particularly from those PSSM components, was also very significant, really showcasing its critical role again.
Okay, sequence is big, evolution's also really important. Did it get even more specific? Like, what about the sequence or evolution matters?
Yes, and this is truly fascinating. The SHA analysis let them drill down. They found that the evolutionary conservation of specific types of amino acid pairs, hydrophobic pairs, seems particularly important for distinguishing allergens.
Hydrophobic, like water-avoiding parts.
Exactly. Think of them as maybe the sticky parts of a protein that help it fold into its specific 3D shape. And that shape is often crucial for its allergenic properties. But here's where it gets really cool, I think. The model actually found known allergenic regions within proteins completely on its own.
It rediscovered known science.
It did. It identified specific motifs. These are short, recurring patterns in the protein sequences, like KMFEGA and IPR-LFK. These exact patterns, or very similar ones, had previously been identified experimentally as IgE binding sites.
IgE. Those are the antibody flags for allergies, right?
The very ones. They're called epitopes, the precise molecular flags that trigger the allergic immune response. And these motifs, the model found, matched known epitopes from common allergens like BETV2 and BETV1, which is the major birch pollen allergen.
Wow. So it's not just predicting based on abstract patterns. It's actually learning and highlighting biologically relevant, known allergenic hotspots. That's huge validation.
It's incredible strong validation. It shows the model is learning real significant biology, not just statistical correlations.
Amazing. Did it identify any other sort of surprising regions or characteristics?
Yeah, one other interesting point was that it seemed to find that the first 200 amino acids of the protein sequences contain a lot of key information for allergen prediction.
The beginning of the protein.
That initial segment, yes. It suggests that region is particularly information-rich for the model's predictions.
Okay, this Multimodal-AlgPro sounds like a monumental leap forward, really. Not just for allergy research, but potentially beyond. So let's talk implications. What does this all mean? Why does this deep dive matter to, our listener?
Well, first and foremost, it achieves state-of-the-art performance. That's key. It doesn't just work. It demonstrably outperforms other leading computational methods. They hit a peak accuracy of 93.9% on independent test sets.
93.9, even better. So that means more reliable, more trustworthy predictions, which is obviously essential when you're dealing with something as sensitive as allergies.
Absolutely crucial.
And reliability is key, right? Especially when dealing with real-world data, which is often messy. Does it hold up okay when the data isn't perfectly balanced? Sometimes you have way fewer examples of allergens than non-allergens.
That's a great point, and yes, it does. It demonstrated strong performance even on these imbalanced data sets. That really showcases its practical applicability. It suggests it could work well in real-world scenarios where data isn't always neat and tidy, which is, let's be honest, a common challenge in biological research.
Okay, practical and accurate.
Yeah.
So who stands to benefit most from this powerful new technology? How might we actually see it applied out there in the real world?
The applications are incredibly broad, really. In public health, for instance, it means better and faster identification of allergenic proteins. That leads directly to improved public safety, allowing for better preventative measures.
Okay, public safety. What about industry?
Big benefits for industry. Think pharmaceuticals, cosmetics, food production. They could use this to assess potential allergens in new products before they even think about hitting the market, saving huge amounts of time, resources, and potentially preventing allergic reactions down the line.
Proactive assessment, yeah. And for researchers themselves.
And definitely for scientific research. It offers novel insights for things like epitope design.
Which is like designing safer proteins or vaccines.
Exactly, creating safer versions of proteins or even designing components for new allergy vaccines or treatments. It can also really help with protein engineering in general and just understanding allergic responses at a much deeper level. Ultimately, it could significantly accelerate scientific advancements across all these areas.
That's a truly significant potential impact across multiple fields. It sounds amazing. Are there any limitations right now or, future directions for this model? What's next on the horizon for this kind of research?
Yeah, like any advanced model, there are always areas for future refinement. One key thing is that currently, Multimodal-AlgPro doesn't fully account for a protein's local 3D structure.
Its actual shape in space.
Its exact geometric shape, yeah.
Oh.
And that shape is absolutely crucial for how a protein interacts with the immune system, how antibodies bind to it. So incorporating detailed 3D structural information is probably the next big step. It could significantly boost performance and accuracy, especially for things like pinpointing those epitopes really precisely.
Okay, adding the 3D shape. Anything else?
Well, there's also an ongoing need for even deeper multimodal interpretability.
More understanding of the why.
Exactly. Not just understanding how individual features contribute, but digging into how the different types of data sequence, evolution, properties, how they interact to influence the predictions. Making the AI more transparent, more trustworthy, that's always essential, especially for broad application in these complex real-world scenarios.
Right, understanding the interactions. Okay, so to quickly recap then, Multimodal-AlgPro, it represents a really significant step forward. It uses this multimodal deep learning approach.
Leveraging diverse protein information sequence, physicochemical properties, and that crucial evolutionary history.
All for superior allergen prediction.
And its key strengths really are that high accuracy, its robust performance even with tricky, imbalanced data, and importantly, its ability to provide interpretable insights. It even rediscovered known allergenic motifs.
Which all points to its real-world value, right?
Yeah.
The potential to significantly advance public health, help industry, speed up research, basically making allergen identification faster, more accurate, and hopefully more accessible.
Precisely. Now, here's a final thought to maybe leave you with. If we can predict allergens this accurately now, what if this technology could be refined further? Imagine if it could predict a specific person's individual allergic risk to any new substance they might encounter.
Wow, personalized prediction.
Personalized allergy prevention as a potential reality someday. All thanks to AI diving deep into this incredibly intricate world of proteins.
What a thought to leave us with indeed. That's fascinating stuff. Thank you so much for walking us through that, and thank you all for joining us on this deep dive. See you in our next episode of the Biosynapse podcast.